Good morning everyone. Today we will be discussing about the history of industrial microbiology. So firstly let's discuss about the microbiology. Firstly, microbiology is the branch of biology that deals with the study of organisms or agents that are too small to be seen by an unaided eye. Then next, basically microbiology has two types of aspects. First one is the basic aspect and the second one is the applied aspect. The basic aspects basically deals with the biology. It is basically the study of biology of microorganism, different microorganisms, like study of bacteria is known as the bacteriology. Then virology. Virology is basically the study of viruses. The next one is the algology, that is the study of algae. Then protozoology, that is the study of protozoas. And the mycology, that is the study of fungi. The applied aspects, they are basically concerned with all the issues like wastewater treatment, then treatment of different disorders and diseases, and food production and food spoilage, and also the industrial uses of microbes. And these both basic and applied aspects, they both are intertwined, that is they both are interlinked, because basic aspect, the applied part is carried out in the basic field, and the application they are basically generate out from the basic research itself. So next industrial microbiology. Thus we can say that industrial microbiology or industrial microbiology, it is an applied field of microbiology, that is it is an applied microbiology that deals with the large scale production of all the economically valuable compounds, or we can say that it is the basically it is the invention or management and exploitation of different microorganisms for the production of different commercially economically valuable compounds. Now this basically the history of microbiology. Industrial microbiology involves 5 different phases, and they are as follows. The first phase is the alcoholic fermentation phase. And it was the period that was before the 1900 era. Then second, after this phase, next phase which came into existence was the antibiotic phase. And it was from the 1900 to 1940. And after that, next phase was the single cell protein phase, or we can say SCP phase. And the duration of this phase was from 1940 to 1964. After this, the next phase was the metabolite phase, or the metabolite production phase. And the duration of this phase was from the year 1964 to 1979. And the last phase of the history of industrial microbiology is the biotechnological phase, or the RDT phase. and started from the year 1980, that is from 1979 onwards. So let's come to the alcoholic fermentation phase. During this era, a lot of discoveries were made, and the major inventions which took place during this era were the production processes for vinegar, beer, and different solvents, and like organic solvents which included citric acid, acetic acid, lactic acid, and butanol. So during this era, firstly the Liebig was, there was one scientist Liebig. He considered as the fermentation process as an chemical process. So he stated that the fermentation process is basically the chemical process, and he gave the conclusion that the different chemical substrates or the different chemical molecules which were involved in the fermentation process, for example sugar. So the he proved that the sugar molecule was responsible for the conversion of sugar into the alcohol. So he considered the chemical molecules as the basis of the fermentation. After him, the Schwann and certain other scientists, they disproved this theory, and they proved that there was the unicellular fungi, that is yeast, which was responsible for the fermentation process. Then afterwards, the Louis Pasteur came into existence, and he finally proved the whole world that the fermentation process is basically an anaerobic process, and it is basically a biological process. In the year 1857, basically he started working on the different on the yeast. So what he found was, so while working with the yeast, he found that different organisms were responsible for carrying out the fermentation of yeast, that is in sugar into alcohol. So what he found was apart from the yeast, there were certain other bacteria also that led to the production of lactic acid. For example, the bacteria Bacillus, Lactobacillus, Lactobulgaricus. And from this theory, he proved that the fermentation process is basically the biological process, that is involving all the biologically important microorganisms. And then from this he gave another conclusion that different fermentations, they require different kind of microorganisms. For example, the lactic acid production involves bacteria, and the alcoholic fermentation, it basically requires the yeast. Then after this work in 1861, Louis Pasteur, while working on the butyric acid fermentation, he made another important discovery while working on the butyric acid fermentation. The important discovery which he made during this work was that the fermentation process is an anaerobic process. That is, it takes place in the absence of oxygen. So during this discovery, he observed certain different microorganisms which were involved. What he found was, during this study, what he found was that there were certain microorganisms which were still active even in the absence of oxygen, and these were basically the rod shaped bacterium, the butyric acid bacterium. He called them butyric acid bacterium. So by this discovery, he concluded that the fermentation process is an anaerobic process, that is, it occurs in the absence of oxygen. And secondly, he concluded that microorganisms, they can be classified into two types of categories, that is the aerobic microorganisms and secondly anaerobic microorganisms. Then during this work, during this phase, he was also asked to give another discovery, he made another important discovery of pasteurization. So during this era, the wine industry of France, it was undergoing a high loss in its due to the soaring of the wine. They were observing the soaring of wine, which caused the heavy loss in the production of wine. So they called up the Louis Pasteur and asked him to find the solution for this problem. So while working on this production of wine, he found that there were certain microorganisms other than yeast that were active during this process, and these microorganisms, they were changing the physical and chemical properties of the wine. So that led to the soaring of wine. So he gave the solution that by partially sterilizing the juice from which the wine was being produced, so by partially sterilizing the wine below its boiling point, we can eliminate all these microorganisms. So he found the solution to this problem and developed a new process which was termed as pasteurization. This is how pasteurization process came into existence. Basically, pasteurization is a process in which all the bacteria, the extra microorganisms that cause the soaring of wine or any other product, fermented product, so they all are eliminated, they become inactive, but it does not change the physical and chemical properties or the quality of the product. During this era, in the year 1684, Leeuwenhoek observed he observed the production of acetic acid, and in the same era, in the year 1861, Louis Pasteur developed another technique, that was Louis Pasteur's effect, which was particularly known as Pasteur's effect. Pasteur's effect is the phenomena in which the physiology and growth of microorganism differs with the conditions, that is whether they are grown in the aerobic conditions or anaerobic conditions. So basically, and they all differ from different microorganisms, there are different growth pattern observed, and what he observed during the Pasteur effect was that less amount of sugar was converted in the anaerobic condition, less amount of sugar was converted into alcohol, and a high growth was observed. And in the aerobic condition, the opposite condition was observed, that the higher amount of growth was observed and the lesser amount of alcohol was being converted into the sugar was converted into the alcohol, that is in the presence of oxygen, the presence of oxygen led in the production of certain other yeast cellular entities. So he finally concluded that in aerobic condition, that is in the absence of oxygen, the less growth occurred and the large amount of sugar was getting converted into alcohol, and in contrast to the anaerobic condition, that is an aerobic condition, the lesser amount of sugar was getting converted into alcohol and a large portion was getting converted into the yeast cellular entity. Next phase, which was the antibiotic period or the antibiotic phase, that was from the duration 1900 to 1940. So the major inventions made during this period were the production of antibiotics, different antibiotics, which included penicillin, then cyclosporine and chloramphenicol. Cyclosporine is basically the drug, and then different apart from penicillin and then other antibiotics which were generated during this phase were the streptomycin, kanamycin, tetracycline and chloramphenicol. And apart from the major advances in the production of antibiotics, certain other inventions were also made, that is the fed batch culture technique was developed during this phase, and another important discovery was the mass production of Baker's yeast and other solvents. So, and basically the penicillin, the different antibiotics which were generated along with the discoverer are as follows. And they are producing microorganisms. So, first was the penicillin. and it was developed by Alexander Fleming in the year 1929. And the causative, the producing microorganism was Penicillium chrysogenum. Then after that, streptomycin was produced, which was followed by streptomycin, and it was developed by S.A. Waxman and et al. And his co-workers. And this streptomycin, this antibiotic, it was produced in the year 1944. From Bacillus licheniformis. Then erythromycin. Before this, chloramphenicol was produced, and it was developed in the year 1947, and it was basically developed by Clark from Streptomyces venezuelae. The producing microorganism was Streptomyces venezuelae, and the last one was the erythromycin, which was developed in 1954 from Streptomyces erythraeus. So erythromycin was developed by Clark from Streptomyces erythraeus. And the penicillin was basically it was required for the treatment of wounded soldiers during the World War II. It was in higher demand, and because it acted as an antibiotic during that time for healing the wounds of the soldiers. Then after that, Weissman, he developed the acetone butanol fermentation process, which was further used for the acetone fermentation also. Then after this phase, next phase was the single cell protein phase. So, and during this phase, it led to the microbial biomass production of proteinaceous material, that is the single cell protein. And the another important invention made during this period was the continuous fed batch fermentation process. And during the last phase, that was the antibiotic phase, after the production of antibiotics, certain other important compounds they were also produced, that were like vitamins, amino acids. Apart from the antibiotics, the vitamins, amino acids and different steroids, they were also being generated during this phase. Then the after the single cell protein, the next phase that followed was the metabolite production phase, that was the fourth phase, and the duration of this phase was from 1964 to 1979. So during this phase, different microbial biopolymers like dextran and xanthan, they were generated, and the techniques for different cell and enzyme immobilization, so different techniques for immobilization of cells and enzymes, that was they were also developed, and the different advanced techniques for the amino acid production, they were also generated. Basically dextran and xanthan, they are secondary metabolites, that is they are not essentially required for the growth and development of any cell. So the dextran, basically it involves its application in the eye drops. It is basically used as an lubricant in eye drops, and it is also used in the increasing the blood sugar levels, and sometimes it is used as an animal feed. Whereas xanthan, another secondary metabolite, it is used as an flavoring agent, and it is used in different items like beverages, different beverage products and ice creams also, and sometimes in sauces also. Now other important discovery during this phase was the included the use of microorganisms for the tertiary oil recovery. So this was the important invention during this period. Now after this metabolite production period, the last phase, the fifth phase was the biotechnological phase, and it was from the 1979 onwards. That is, after the 1979 all the inventions made were during this period, and the rapid progress in the development of industrial microbiology basically led to all these things. And main processes due to which this rapid progress was possible in the industrial microbiology was the development of two different techniques. First one was the recombinant DNA technology, or the RDT, and the second one was the development of the monoclonal antibodies by hybridoma technology. So, these two played a really important role in the development of an industrial microbiology, and different hormones were developed during this era, and different vaccines were developed. For example, in the year 1982 recombinant hepatitis B vaccine was developed, and certain other vaccines were also developed. And the basically the hormones included insulin, human insulin, which was used in the treatment of diabetes, and it was followed by the production of another human growth hormone, HGH, and it was further followed by the development of CSFs, that is the myeloid colony stimulating factor, which is used in controlling the production of RBCs, that is the red blood cells, by stimulating the proliferation. Then after this, after the CSFs, this production was followed by lovastatin production, that is the somatostatin, another human growth hormone. This was developed followed by the development of erythromycin, which is used for treating the anemic patients, and it is also used in the renal failure, treatment of renal failure. Then after this erythropoietin, it was the protein erythropoietin. All these were developed from the recombinant DNA E. coli. Erythropoietin was followed by the discovery of GCSFs, that is the gametocyte colony stimulating factor, involved in treating different type of cancers. Then another important discovery made during this period was the production of production of microbial secondary metabolites. For example, there are certain like chloramphenicol, which is a drug, and then another important secondary metabolites involved lovastatin and imipenem, lovastatin, then cyclosporine. It is an immunoregulant which is used for the different type of organ transplantations. And after lovastatin, there was another important drug, ivermectin. Lovastatin is a broad range antibiotic. Ivermectin is an anti-parasitic drug used in the treatment of African blindness disease, and the imipenem, it is also the lovastatin, it is the drug used in the for reducing the blood cholesterol level. Thank you.